All right, another technique that's really powerful for use in the biotechnology laboratory, it's used on almost every crime show, and that's the polymerase chain reaction. The polymerase chain reaction is a method that was invented in the mid-1980s that revolutionized molecular biology by allowing scientists to specifically amplify. And when I say amplify, I mean make millions of copies, excuse me, make millions of copies of a desired sequence. So we can actually see how that happens in this process here. If we look at a DNA molecule, we might decide, for example, I have a lot of DNA floating around, but here's the one DNA that I want. And so what I'm not showing in this figure are the millions of copies of DNA that I don't want, okay? So maybe I have a DNA from a crime scene, for example, and I have a region of DNA that I know is unique to an individual that is a suspect in the crime. I only want to amplify this DNA and I don't want to amplify all of the other sequences that are there. My question is, can I amplify that? Well, using the polymerase chain reaction, I can. To do that requires some special setup, and the setup involves some chemicals that I need to use and a machine. So let's talk about those. When we talked about DNA replication, we talked about that DNA polymerases all require a primer. And in the cell, a primer consists of an RNA that's made by an enzyme called primase. In the technique I'm getting ready to describe to you, the primer that we use is not RNA, and in fact, it's a chemically made DNA that's short in sequence, about 20 nucleotides or so, and it's complementary to a portion of the sequence I'm trying to amplify. Now, there are two primers that are made. One primer that comes from one end of the DNA and is complementary to one strand, and the other primer that comes from the other end of the DNA that is complementary to the other strand. Now, that means that the process I'm getting ready to describe means I first of all have to have knowledge of the sequence to be amplified. Second, I need to make two primers. One complementary to one end, one strand, the other complementary to the other end, the other strand. So now I have the two primers. The primers are important because the polymerase requires the primers. It will not start replication without them. The second thing I need to have is I need to have dNTPs because dNTPs are needed for making DNA. And the third thing I need to have is a DNA polymerase that will put those together and copy that DNA. If I have those things, in addition, of course, to the DNA that I want to amplify, I can do this process, and the process is really magical. So the screen shows how this process occurs. The first step in the process involves separating the strands, and that's a process called denaturation. In denaturation, I use boiling water to separate the strands, and when I do that, the strands come apart. The second part of the process involves cooling the mixture down to a temperature where the complementary primers will find their respective strands. And they literally find them and form base pairs like what you see here. The third part of the process involves the DNA polymerase starting at the primer and replicating the desired sequences. Now notice what I've done in the process. I've started with two strands, and by the time I get down here, I have 4 strands of that same DNA. If I repeat the process, these 4 strands will become 8 strands. And if I repeat the process again, those 8 strands will become 16, so that I have a theoretical doubling each time I run through the 3 steps of the process. That theoretical doubling, okay, can give rise to as much as 1 billion times as much of this as I started with if I go through only 30 cycles. Now the last thing, that's first of all, very incredibly powerful, I guess I want to say. But the other thing I want to point out is a very clever idea that was built into the method when it was first designed, and that was to use a DNA polymerase, okay, that was not sensitive to heat. Most enzymes, including most DNA polymerases, will be destroyed by boiling them. So every time I boil, I would have to add a new polymerase if I wanted to continue using the technique. Well, people went out and investigated bacteria that live in hot environments. And the one they found was something called Thermos Aquaticus, and it lives in Yellowstone's Old Faithful geyser, and it has a DNA polymerase that doesn't get destroyed by boiling. So one can do this whole process in the same tube without having to add a new DNA polymerase each time.